Body Fluids and Circulation, Session 2. In this module, we are going to learn about blood grouping. What is your blood group? A, B, AB, or O, which is either positive or negative. Let us learn in detail about how we obtain this blood group. The blood group is also called as a blood type, and the classification of this blood group is based on whether the RBC surface is having an antigenic sugar called antigen A or antigen B. In the first picture of the RBC, we can see that the surface is having certain antigenic sugars, whereas in the second picture, the RBC surface is devoid of any antigen, and that is the basis of classification of blood group. The blood grouping system was discovered by Karl Landsteiner, who is a biologist, physician, and an immunologist. And he discovered two types of blood groups in humans. The two famous blood groups are ABO blood groups and RH blood groups. ABO blood group is based on the presence of two antigens, namely antigen A and antigen B. And RH blood group is based on the presence of an antigen, RH antigen. There are four types of blood groups in ABO blood grouping system: A, B, AB and O. In persons with A blood group, the surface of the RBC is having the antigen A. People with B blood group have the antigen B on the surface of the RBC. People who are having AB blood group, on the surface of the RBC, they have both antigen A and B. And O blood group are devoid of any type of antigens on their surface. So once again I repeat, blood group A have antigen A, blood group B have antigen B, blood group AB have both antigens A and B, and blood group O are devoid of any type of antigens. In addition to antigens, antibody are also present in the blood. In persons with A blood group, they have antigen A on the RBC and antibody B is present in the plasma. Anti-B means antibody B. That means the antibody which acts against antigen B. In people with B blood group, they have antigen B on the RBC surface and antibody A in the plasma. Anti-A means against antigen A. AB blood group have both antigen A and B on the surface of RBC and they lack any type of antibodies. Neither antibody A or B is present in their plasma. Whereas O blood group, we cannot find any type of antigen on the surface of RBC. Whereas both antibody A and antibody B are present in the blood plasma. So this is the distribution of the antigen and antibodies in different blood groups. At the time of blood transfusion, it is very important to do the blood matching or blood compatibility between the donor and the recipient. We know donor is a person who donates blood and the recipient is a person who receives the blood. If there is any type of mismatch between the donor and the recipient, there are chances of antigen antibody reaction. And because of this antigen antibody reaction, agglutinization of the RBCs takes place and this will result in a dangerous condition. So it is very important that for blood transfusion, there should be a blood group matching between the donor blood and the recipient blood. We have already discussed that people with A blood group have antigen A on the surface of RBC and in their plasma, antibody B is present. Antibody B means it acts against B antigen. And during transfusion, if B blood group is injected or transfused into the recipient having A blood group, immediately the RBCs of the injected blood or the donor's blood will start to be destroyed by the antibodies which is produced in the recipient, that is A blood group. In the same way, if the donor is an A blood group and the recipient is a B blood group, the recipient will have antibodies A, and when the A blood group enters into the recipient, the RBCs will be destroyed by the antibodies of the recipient. O blood group are called as universal donors. Let us understand why. Universal donor means they can donate blood to any type of blood group A, B, AB, or O. It is because people with this type of blood group, they do not have any type of antigen. Antigen A or antigen B is absent on their surface, and so they can be safely given to any people of any type of blood groups. Whereas AB blood group are called as universal recipients. We know AB blood group have antigen A and B, whereas they lack antibodies. Antibody A and antibody B are absent in AB blood group. So they can receive blood from any type of blood group, and hence they are called as universal recipients. Next, we will learn about the RH blood group. The RH blood group is the second important blood group in humans after ABO blood group, and it was discovered by Karl Landsteiner, and it is based on the presence of RH antigen or the absence of RH antigen. If the RH antigen is present on the surface of RBC, then it is called as RH positive. Whereas if the RBC lack the RH antigen, then it is called as RH negative. Dear children, do you know what is the meaning of this RH? RH is an abbreviation of rhesus factor. It is an abbreviation of rhesus factor. It is called as rhesus factor because it was similar to the antigen found in the RBCs of rhesus monkey. Hence, it is called as the rhesus factor. Rhesus monkeys RBC had the similar type of rhesus factor, and which is found in the humans, and those who have this RH antigen are called as RH positive, and who lack the RH antigen are called as RH negative. Dear children, in this module we learnt about the two types of blood groups in humans, ABO and RH blood group. Thank you.